Okay. All right, so a week from today, we have midterm number 2. We have our second midterm in 1 week. And we're doing well in terms of the pacing of lecture material. We're ahead of schedule. And so what that means is that we can dedicate the full class period on Monday to a review session. So we'll run it the same way as last time. You guys bring questions, I'll answer them. And if you don't want to attend the review session, we won't have any clicker questions. You don't have to show up if you don't want to. Okay, so we'll have that on Monday. We'll have the test on Wednesday, and then we'll resume these Part 3 lectures that we've been focused on. And so today what we're going to do is we're going to continue on that. We have one more example of a reflex to talk about, and then I'll change slides onto the next topic, which is on energy balance. All right, so we laid out all these efferent pathways. These are the various ways that your central nervous system can recruit the organs of the body to do something about a state variable, something where it wants to maintain homeostasis. And we talked about somatic pathway last time. That was the patellar tendon reflex, where the effector organ was a skeletal muscle. We're conscious of that because of skeletal muscles. Whereas there are a variety of reflexes that you're not conscious of. And we're going to consider one that uses a parasympathetic efferent pathway. And that is the micturition reflex, or the urination reflex. And so we talked about the kidney and how the kidney works and nephron and all that kind of stuff. So when the filtrate that emerges from the collecting duct of a nephron is passed on into this tube that's connected to the urinary bladder, the production of urine by the kidneys inflates this muscular sac, the urinary bladder. And so let's consider the reflex action for urination. That is the clearing of urine from the urinary bladder. And just like for all the control systems, we're going to want to lay out its basic parts. We're going to have sensors, integration centers, and efferent pathways. And so our stimulus for this control system is the tension that emerges in the walls of the urinary bladder. So there are afferent neurons that are similar to the muscle spindle organs we talked about for the patellar tendon reflex that were in your quadriceps muscle. But these mechanoreceptive neurons are just woven into the walls of the urinary bladder. And so they are sensitive to the stretching of the urinary bladder. I don't know if talking about this will make you feel like you have the urge to urinate, but just imagine the tension in the walls of the urinary bladder, the stretching of the dendrites of those sensory cells. They pass through the dorsal root ganglion, right, that thickening just outside of the spinal cord, and they terminate in the gray matter. And it's in that region that there will be a chemical synapse, which I'll explain in a moment. But so we've added that stretch receptor that is sensitive to the bladder tension. And again, the stimulus, the sign of the variable matters. This is a heightened bladder tension that excites the stretch receptors. Then, because of the termination of that neuron in the gray matter, we have the passing of that signal to the spinal cord. And it's in the spinal cord that decisions are made about the efferent responses. And in particular, it's a slightly more complex series of decisions because there are more chemical synapses that are involved. Now, this being a parasympathetic pathway means that the neuron that is activated has a synapse along the way. But in between, there's an interneuron here in the gray matter. All right, so the plus here means that when there's an excitatory signal that initiates from the blue, that is the afferent neuron, and then the interneuron transmits that signal, that has an excitatory effect on this neuron here, which terminates in the smooth muscle that lines the urinary bladder. Okay, so if there's more wall tension in the bladder as a consequence of filling up with urine, then the response here is to increase the tension of the smooth muscle lining of those walls. Okay, and so that is via a parasympathetic pair of neurons that are having that positive effect on the smooth neuron. Now this would be, we talked about in the last case, the example of a monosynaptic reflex because there was only one synapse between the muscle spindle organ and the motor neuron of the quadriceps muscle. Here we've got a polysynaptic reflex. Okay, there are two chemical synapses, one there and one there, and then along the way we also happen to have this one as well. Okay, the afferent neuron also branches, and this is another way it's a little bit more complex. You wouldn't include this branch in deciding whether or not it's mono or polysynaptic, but this branch is going to activate another neuron, another efferent neuron in parallel. It again has an interneuron in between, but that interneuron is inhibitory. Okay, so what is that other neuron? Well, we can see here that that second neuron has no interneuron, and in fact, it terminates onto a skeletal muscle. Aha, so this is one we might be conscious of and be able to exert some conscious control over. That skeletal muscle is wrapped around or provides the basis for a sphincter, a closure of the urinary bladder. So we're going to see in this case and in other examples that we have neurons that spontaneously generate a train of action potentials. Okay, it's not just when they receive an excitatory stimulus, they just are capable of a basal rate of action potentials. And this is an example of that. So in other words, the motor neuron, that is the one with its cell body as the lower cell body there, or the one at the left, that motor neuron is always maintaining some level of tension on the skeletal muscles by sending it a series of low-frequency action potentials. All right, that's what it does. And so when I say train of action potentials maintains tension, that's what I mean. Just imagine there are these periodic action potentials that are keeping that sphincter from opening because there's a maintenance of tension that keeps it closed. So, that being the case, when there's tension in the walls that are sensed by these dendrites, an excitatory signal that's passed down here, then this interneuron, which has a negative effect on the motor neuron that's maintaining that closure of the sphincter, when that's activated, that means it's going to cause a depression in the firing rate of that motor neuron. In other words, that's going to allow for a relaxation of the sphincter. So we add that to the control system. In addition to enhancing the tension in the walls of the urinary bladder by contracting the smooth muscles of those walls, there's also an inhibition of the skeletal muscle that keeps the urine on the inside. So as a consequence of both of those parallel actions, we get urination, the emergence of urine from the urinary bladder. And so that is the ultimate efferent response. Okay, again, this is initiated by a stimulus, and then all the actions just play out. So the stimulus does not feed back onto the efferent response. And so this would not be an example of negative feedback so much as it is a feed-forward situation. Okay, and this is known as the micturition reflex, and as I just said, it is a feed-forward control system where the response of the autonomic pathway and the somatic pathway does not vary with the afferent input. All right, and that is what we are born with. When our urinary bladders fill up as infants, then we just pee. We just release that tension in the walls. The skeletal muscle sphincter that provides a closure for the urinary bladder just relaxes, and we wet our pants. But fortunately, we learn to control this reflex. And so how is that? How do you modulate a reflex action so that we can exert some control? So this can be inhibited by descending commands. Okay, it is natural to just wet yourself whenever your bladder fills up. But because the brain can exert some control over this, then we can influence the timing on when the urine is released. And that is through descending commands. So through the white matter of your spinal cord, you have these long cells, these long neurons with their cell bodies up in your brain, and they issue along a long axon descending commands. And in the case of this reflex, we have a descending command that influences the autonomic pathway. And that would be, let's see, a negative effect. So did I fit? I didn't actually fit a negative in here. But just imagine on the right synapse, there is a dendrite on that parasympathetic neuron, that is the one on the right, receives a negative signal from the brain. So we can add that to our control system. And what that's going to do is inhibit the positive effect on the smooth muscle. So way back when we were talking about how neurons work or how dendrites integrate signals, and we were sort of describing that in the abstract. Here again, we see a nice example of that, where you've got some inhibitory synapses, in this case, that come from the brain, which tend to hyperpolarize the membrane of that parasympathetic neuron, so that it makes it less likely that the excitatory stimulus coming from the afferent side, that is those the ones that are shown in blue here, they are less likely to trigger the contraction of that smooth muscle wall. Yeah?
So we can control an autonomic pathway through descending pathways?
Yeah, the question is, can we control an autonomic pathway through descending pathways? Yes. Over long periods of time, it is possible for some bodily functions to be controlled.
So can we control our heartbeat then?
So can you control your heartbeat like a ninja? Or, you know. So I think that's a subject of debate. So theoretically, we see, yes, physiologically, it is possible to modulate autonomic nervous function. But whether or not it's actually possible for certain organ systems, in the case of the heart rate, I don't know. I do know that it is possible by conscious action to, meditate or other ways of relaxing yourself, and then they secondarily can influence the autonomic nervous system. So when we went through that elaborate table of all of the parasympathetic and sympathetic pathways, those can all be modulated by visualizing certain events. You go to a happy place in your brain, or just going to the beach or something like that. They are more likely to lend to parasympathetic activity when you are relaxed, however you can manage to relax yourself. Whereas if you perceive something as a threat, even if it isn't a threat, then you're much more likely to be activating the sympathetic pathway. So there's certainly plenty of indirect ways. Here we see an example of a very direct way that we can control an autonomic pathway. Sure. So not only do descending commands have the capacity to influence the autonomic side of things, but we also can send descending commands to do something about that sphincter. And that's a more familiar situation for us at this point because of the skeletal muscle that's involved. And again, it's a negative effect. So just imagine a negative chemical synapse, chloride ions, etc., influencing the cell body on the left there. And so that descending command is going to negatively influence the negative effect of the motor on the skeletal muscle. So here we have an example of a double negative because you may recall a moment ago that interneuron that's shown on the left was negatively influencing the motor neuron for the skeletal muscles. That was so that the skeletal muscle relaxes when your urinary bladder is full. Here, we're turning down that negative effect. So as a consequence, the high frequency of action potentials is maintained for that skeletal muscle, and the tension of that skeletal muscle stays elevated. In other words, it doesn't open up, and as a consequence, you don't pee when you're receiving these descending commands. All right, so the micturition reflex can be inhibited by these descending commands that emerge from the brain. And there are also sympathetic inputs to this reflex as well. So we touched on that when we did lay out that table. So for the urinary bladder, we just talked about how we have, from the parasympathetic pathway, an enhancement of urination. The walls of the smooth muscle lining of the urinary bladder also has alpha and beta 2 receptors. And if there are alpha receptors, then you can infer that there are going to be sympathetic neurons that release norepinephrine. And so it's possible for the sympathetic nervous system to influence bladder tension as well. Basically, if you're being chased by a bear, then the walls of the urinary bladder tend to relax. You are less focused on urination in general. You might ask the question, well, what about people who are so scared that they pee their pants? I don't really have the answer to that, except for there seems to be a very nonlinear effect of sympathetic inputs. Okay, any questions about micturition? Yeah, question?
I got a question about the receptor. So based on what I'm seeing right now, is the receptor only applied to sympathetic, or it could apply to both parasympathetic?
No, so those receptors, so here the color coding matters. I apologize for those who are colorblind because we got green and red here. But so the sympathetic is shown in green, and then the receptor is also shown in green. And those receptors only refer to sympathetic. They're only responsive to either norepinephrine or epinephrine, those particular receptors. Parasympathetic inputs have no influence on the activity of alpha and beta receptors.
Thank you.
Mm-hmm. Any other questions? Okay, I'm going to switch my slides. Just give me a second here.